Welcome to this lecture on recombinant insulin and growth hormone. Today, we are going to explore one of the most monumental achievements in animal biotechnology and molecular medicine. We will learn how scientists have harnessed the universal nature of DNA to program simple bacteria, turning them into microscopic factories that churn out complex human proteins. This technology has saved countless lives, and by the end of this session, you will understand exactly how it works, both biologically and mathematically. To begin, let us imagine you have a friend who desperately needs a rare replacement part for a vintage car. If the manufacturer stops making it, you could take the blueprint to a 3D printing factory to make perfect copies. This is exactly what we do when the human body fails to produce essential hormones, like insulin or growth hormone. Instead of relying on a broken biological system, we take the human genetic blueprint and insert it into microscopic factories, usually bacteria or yeast. These tiny cells read the blueprint and manufacture the exact human hormone in massive quantities, allowing millions to live healthy lives. Now that we understand the basic idea of microscopic factories, let us look at the formal biological process. This relies on recombinant DNA technology. First, scientists isolate the specific human gene, such as the INS gene for insulin or the GH1 gene for growth hormone. This gene is spliced into a circular piece of bacterial DNA known as a plasmid. We then introduce this recombinant plasmid into a rapidly dividing host, most commonly the bacterium Escherichia coli. Because the genetic code is universal across all life forms, the bacteria read the human gene as their own, synthesizing the human protein. Finally, we extract and heavily purify these proteins to create safe, injectable medicines. Understanding how we make these proteins is fascinating. But why did we need to invent this technology in the first place? Before recombinant insulin was approved in 1982, diabetic patients injected insulin extracted from the pancreases of slaughtered pigs and cows. While it worked, it often caused severe allergic reactions. Similarly, human growth hormone was painstakingly extracted from the pituitary glands of human cadavers, a dangerous process that tragically transmitted fatal brain diseases like Creutzfeldt-Jakob disease. By shifting to recombinant biotechnology, we now produce these hormones in completely sterile, controlled bioreactors. This guarantees an unlimited, perfectly safe, and completely human-identical supply, preventing severe complications and early death for millions. Before we move on to the mathematics of scaling this up, let us visualize the entire biological process we just discussed. On the left side of this diagram, you can see the human cell providing the specific gene, and the bacterial cell providing the circular plasmid. Notice how restriction enzymes act like molecular scissors to combine them into a single recombinant DNA molecule. This new blueprint is then inserted back into an E. coli bacterium through a process called transformation. The bacterium is placed into a large bioreactor, shown on the right, where it multiplies rapidly. Finally, the product is extracted and purified, ending up in the sterile medical vial you see at the far right. This is the complete journey from genetic blueprint to life-saving medicine. Now that we have seen the biological process, we must ask, how do we produce enough of these hormones for millions of people? This brings us to the mathematics of bioprocessing. We calculate the total yield of a recombinant protein using a fundamental biomanufacturing equation. Y equals C times V times E times R. Let us break down these symbols. Y is the final yield of the purified hormone, usually in grams. C stands for cell density, meaning how many bacterial cells are in 1 liter of broth. V is the total volume of the bioreactor in liters. E denotes the expression level, which is the average mass of protein produced by a single cell. Finally, R is the recovery efficiency, a decimal representing how much protein survives the purification steps. If we increase the bioreactor volume or cell density, our yield goes up. But if our purification process is harsh, R decreases, and we lose valuable medicine. Let us see this formula in action with a concrete example. A pharmaceutical company is using a 5,000-liter bioreactor. They achieve a cell density of 2 × 10 to the power of 13 cells per liter. Each cell expresses 5 × 10 to the negative 14 grams of raw insulin, and the purification efficiency is 60%. We need to find the final yield in grams. We start by writing down our fundamental yield formula. We substitute our given values for volume, cell density, expression level, and recovery rate. Multiplying the cell density by the volume gives us the total number of cells. Multiplying that by the expression level shows that the bacteria produced 5,000 grams of raw insulin. Finally, we multiply by the 0.60 recovery efficiency to account for losses during filtration. The final answer is 3,000 grams, or 3 kilograms, of pure medical-grade insulin, enough to sustain thousands of patients for a year. Now that we have successfully used the yield formula, let us understand exactly where it comes from by breaking down the derivation step by step. The formula is built by tracking the physical mass of the protein at each stage of manufacturing. The very first thing we must establish is the total workforce size. We cannot know the total protein output without knowing how many microscopic factories are working for us. We define N total as the total number of bacterial cells. This is simply the cell density, C, multiplied by the total volume, V. In our previous example, this calculation revealed a staggering 1 × 10 to the power of 17 total bacterial cells actively producing insulin. This massive number is the foundation of industrial fermentation. This brings us to the second stage of our derivation. Once we know the total number of cells, we need to calculate the absolute maximum amount of insulin present in the biological soup before any filtration takes place. We call this the raw mass, denoted as M raw. To find this, we multiply the total number of cells, N total, by the expression level per cell, E. By substituting our previous equation, we see that M raw equals C times V times E. This represents the biological efficiency of our genetically modified bacteria. In our example, this theoretical upper limit was 5,000 grams. However, at this stage, the insulin is mixed with toxic bacterial cell walls and DNA, meaning it is not yet a usable medical product. This leads us to the final step of our derivation. We must account for the inevitable losses that occur when we separate the pure insulin from the toxic bacterial soup. Purification involves multiple harsh stages of centrifugation and chromatography, and at each stage, a small percentage of the precious insulin is destroyed. Therefore, the final usable yield, Y, is the raw mass multiplied by the recovery efficiency, R. When we substitute our expression for the raw mass, we arrive at our complete final derived formula. Y equals C times V times E times R. By breaking it down this way, we can clearly see how biological metrics like cell density and engineering metrics like recovery efficiency combine to determine the final availability of the drug. So we have proven how to calculate yield for a single batch. This brings us to a different challenge. How do we design a facility to meet the needs of a specific population? Let us look at human growth hormone. Before building a bioreactor, engineers must calculate the total annual demand, denoted as D annual. The formula is simple. D annual equals P times D times W. Here, P is the total number of patients relying on the clinic. The variable D represents the specific weekly dose required by each patient, measured in milligrams. Finally, W is the number of weeks in a year. If the patient population increases, or if the required dose goes up, the annual demand scales linearly. Failing to meet this exact target means real patients will go without their necessary treatments. Before we move on to sizing the bioreactor, let us see this demand formula with a concrete example. A clinic needs to treat 500 children with severe growth hormone deficiency. Each child requires exactly 10 milligrams of recombinant human growth hormone per week. We need to find the total mass required for the entire year. We start with our annual demand formula. We substitute 500 for the number of patients, 10 for the weekly dose, and 52 for the number of weeks in a year. Multiplying these together gives us 260,000 milligrams. To make this number more manageable for engineering purposes, we convert it to grams, giving us a final answer of 260 grams. This is the absolute minimum target our manufacturing facility must hit over the course of the year. Now that we have calculated the annual demand, we must translate that long-term yearly goal into a physical engineering specification, the volume of the bioreactor. We do this using a combined formula. V, the minimum working volume in liters, equals the required yield per batch divided by the specific yield per liter. Let us look at the symbols. Y batch is the required yield per single batch, which we find by dividing the annual demand by the number of batches run per year. We then divide that batch target by Y liter, which is the known mass of purified protein we can extract from a single liter of broth. If our specific yield per liter drops, we will be forced to use a much larger bioreactor volume to compensate. Let us apply this to our clinic's needs. We know the annual demand is 260 grams. The facility runs exactly one batch per month, meaning 12 batches a year. The specific purified yield is 0.5 grams per liter. We need to find the minimum working volume of the bioreactor. First, we divide the annual demand of 260 grams by 12 batches. This tells us that each individual batch must successfully produce at least 21.67 grams of purified growth hormone. Next, we take this batch requirement and divide it by our specific yield of 0.5 grams per liter. The final answer is 43.34 liters. In a real-world setting, engineers would round this up to a standard 50-liter bioreactor to provide a crucial safety margin, ensuring the children always receive their medicine. We have covered a tremendous amount of ground today. Let us review the key takeaways. First, we learned that recombinant DNA technology allows us to program simple bacteria to act as microscopic factories, churning out complex human proteins with perfect accuracy. Second, we saw how this breakthrough completely eliminated the dangerous historical reliance on animal organs and human cadavers. Third, we explored the mathematics of biomanufacturing, proving that total yield is a delicate balance of cell density, bioreactor volume, biological expression, and engineering recovery efficiency. Finally, we learned how to scale these systems by calculating annual patient demand to determine the exact physical size of our bioreactors. You now understand how manipulating DNA at the microscopic level can solve massive global health challenges. Thank you for your attention.